guys, and welcome to IV Online. Javed Khan Afridi here, and you are watching AS Level Biology. Today, we will discuss about the cell cycle. What is cell cycle, and why do you need the process of cell cycle? Cell cycle is actually a series of events where one cell, usually known as the stem cell, divide into two daughter cell, and that complete cycle, which is consist of so many steps, constitute the entire process of cell cycle. Now in this chapter we will discuss different changes in the cell during the process of cell cycle. I mean, changes in the size, changes in the genetic and cytoplasmic contents of the cell, and the significance of the daughter cell which are formed as a result of cell cycle. So what is cell cycle and what is its importance? That will be discussed today in this video. So let's discuss some more detail about the cell cycle. You know, in our body there are two types of cell. So we say that cell are of two types. Cell maybe are of two types. One is called stem cell and one is called specialized cell. You know that the stem cell has the potential to divide, multiply. This is the unique aim of the stem cells, to amplify the number of identical cells as the parental stem cell. So the stem cells nothing have to do. They just have to increase the number of similar cells, identical cells, in huge number. So of course, when there's many cells, they will make the tissue, and when the tissue is further growing, they makes the organ and then the organism. So you know, it plays very important in the development, growth of the tissues, of the organ, and even of the organism. So stem cells are those cells which have the potential to multiply, which have the potential to produce identical cells. One cell divide into two, and both cells are not only similar to one another but also similar, identical to the cell from which they have been formed. Now, once the cells are dividing and multiplying, so many times, their potential is gradually decreasing, and once the cell is specialized. How the cell is specialized? The cells perform its unique function, which is different from the rest of other cells. Then we say that the cells become specialized. For example, I'm just giving you a few examples, like one of the stem cells is the bone marrow. So bone marrow cell, bone marrow stem cell, they divide into daughter cell 1 and daughter cell 2, which are both identical to the bone marrow stem cell. Then the daughter cell will again divide into two, then they again divide into two. In the similar way, the number of cells are going to be increased after so many generations if we say that no one is going to be specialized. If no cell is specialized and there just remains the stem cell, so you know, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So you know that even from one bone marrow stem cell, we have produced 16 cells which may be identical to the bone marrow cell. This process remains continued for a long period of time to divide and to produce thousands and thousands and billions of cells by the same way of multiplication, so to increase the number of identical cells. So such cells are called stem cells, which produce the same and identical cells, and the cells are not differentiated from one another. They are just like identical to one another. So stem cells nothing have to do. They just have to increase the number of cells. Now, other kind of cell is the specialized cell. Specialized cell is the one which start its unique function. Suppose if this cells which have been the daughter cell produced from the bone marrow cell is developed into RBC, red blood cells, because it's formed from the bone marrow. Similarly, this cell is modified to form neutrophils, which is a white blood cell. Similarly, this one is specialized to form macrophage, which is also a phagocyte, white blood cells, and so many other cells. So, RBC, neutrophil, and macrophage, they don't have the potential to produce more macrophage or more neutrophil. Means that specialized cells don't have the potential to produce identical cells like the stem cells. So once they perform their functions, then they will die and they will be replaced by other stem cells to produce again more RBCs and neutrophils and macrophage. It means stem cells have the potential to produce from the backward more specialized cells, and specialized cells after performing their functions will be disintegrated, they will be died, and they'll be removed from the body. Like the skin cells on monthly basis or on weekly basis, the skin cells are replaced by the newly formed skin cells just beneath the dermis region. So it means, if you think about that, we say that each and every tissue and organ in our body must possess the stem cells to regenerate the tissue. Otherwise, it will be finished, it will be removed from the body. Like if you are talking about the skin and the skin cells are removed, so it means the skin tissues also have the stem cells to produce the new cells which develop the skin. Similarly, the hair papilla, which multiply to produce hair, and new hair we are shaving and we get new hairs from the bottom, which means stem cells are also present in the hair follicle. Similarly, the bone marrow cells are the stem cells which produce the RBC, white blood cells, and so many type of cells. And RBC, they just remains only for 3 months or 4 months, they become disintegrated, completely removed because they are destroyed by the liver. So the new RBCs are formed again from the stem cells and so on. So we can say, for simplicity, if there's one of the stem cell and it goes to divide into two daughter cell. So this is one of the daughter cell, which is exactly identical to the stem cell. The other one is the daughter cell 2. Now what's going on? Let's suppose that this cell goes to become specialized and it start its function. So once it's specialized, maybe it become the root hair cell. Now, root hair cell cannot produce more root hair cell. As it's specialized, it has different shape, it has adaptation, it has increased surface area, prolonged surface area. The cytoplasm have extension, which is different from other epidermal cell. Root hair cell is an epidermal cell, but it's quite different from the rest of the epidermal cells. And this daughter cell again acting as a stem cell, and it again produce another daughter cell 1 and then the daughter cell 2. Similarly, now this cells become specialized and it again divides into two, daughter cell 1 and daughter cell 2. In this way, the specialized cells will become finished. After performing their function, they have been disappeared. Now they will be replaced by other stem cells, which again differentiated into the in the specialized cell. That's why the number of cells will never be reduced in the body, because they will be replaced by the stem cells. So stem cells regenerate the cells, identical size, identical shape, and the specialized cells are performing their function. Now, we can say that what is the cell cycle. So we say that cells that divide repeatedly to replace those lost. So who are the cells which are lost? These are the specialized cells, because specialized cells are working for a certain period of time and they cannot divide again. They will be directly died. So once the potential of the stem has been finished into specialized cell, the specialized will be then died after performing its function. So they will be replaced by more, stem cells. So from the cell surface like skin, go through a cycle of events called the cell cycle. Now these events eventually allow the cell to divide to form two cells by mitotic division. Mitotic division, identical cell will be formed because the DNA is exactly copied. One of these cells become a specialized cell, not necessarily. Maybe both of them are the stem cells, and in the second generation they may become the specialized, or maybe in the third generation they become specialized. But in some time in the cycle process, some of the cells become specialized, while the rest of them become the stem cell. The other complete the cell cycle so that the process can be repeated. The cell cycle is shown in the flow chart below using a stem cells in the bone marrow as an example. So here are the stem cells in the bone marrow. It divides. Now, cells complete the cycle. So if the cycle is completed, then it makes the daughter cell 1. Cells does not complete the cycle, daughter cell 2, so it will become specialized. So if these cells don't have the potential to complete the cycle, it means it becomes specialized and it remains again the stem cell, and the process will be remain continued. Now, how a cell becomes specialized? Why this cells not become specialized? Why this cell becomes specialized? Now it's like somewhat interesting story. Giving you an example, let me suppose that we have a stem cell. This is the nucleus of the stem cells. This is the nucleus of the stem cell. And this nucleus has so many DNA molecules, many chromosomes. We're just talking about one chromosome, only one DNA, and that is chromosome number 6 or 5 or 7, anyone. So I'm just selecting only one of the chromosome from the nucleus. And I just make some partition here. These are the genes which are located on this chromosome. This is, let's suppose this is chromosome number 6. So here's the chromosome number 6 or maybe 7. Let's say 7, chromosome number 7. We have 23 chromosomes in our nucleus. 46 chromosomes we have in our nucleus. So this is only one chromosome been taken, chromosome number 7. Now it has gene. This is gene A, this is gene B, gene C, gene D, gene E, and gene F. You know that this stem cell is going to divide into 2 daughter cell, and the daughter cell produced, of course, will be identical, and they have the same setup of gene. Again, gene A, gene B, gene C, gene D, gene E, and gene F located like in the same way. And similarly, the other nucleus, the other cell also the same nucleus, have the A, B, C, D, E, and F. Now, in the previous chapter, in the genetic control, I told you about the gene expression. Once a gene expressed, it means it is transcribed and translates to produce a particular protein. Suppose that these cells remain unexpressed to, neither of the genes here expressed. If no one of the gene in daughter cell 1 has been expressed, it means the cell still has the potential to divide again by next cycle. So it will divide again. This is only one chromosome and there are 46 chromosomes. If there is a particular protein required by the cell, and that protein has been formed by the cell, the gene is expressed to form the protein, then the cell will not be divided furthermore and it will become specialized. But suppose the gene C, I'm just going to shade it as here, the gene C. If this gene C is responsible to code. Gene C is responsible to form antibodies. You know, antibodies are protein. And proteins are formed from polypeptide. And you know, polypeptide is coded by a gene, as I told you in the previous chapter. And the gene here is the gene here is C. Now, if the gene C is remain inactive and it does not express its position in daughter cell 1, it means this cell will never produce antibody. They will never transcribe or translate. So the cell does not perform its function. If it does not perform its function, it means it remains as stem cells and it again produce the newly daughter cells. Now what's going on in daughter cell 2, that the gene C, this gene C has been expressed. So once the gene C has been expressed, they will make messenger RNA. Messenger RNA will be translated to form polypeptide, and the polypeptide then protein, and the protein then makes antibody. It means this cell start to secrete antibodies. Now, secretion of antibodies means a function. Now, this cell starts its function. It has the potential to produce antibodies, which is somewhat unique function. There's no other cells to produce antibodies. So once it started its function, now it's become a specialized cell. So D2 is a specialized cell. Now this cell cannot be divided into other similar cells because it now lacks the potential of division. So now this cell does not have the potential to divide, but daughter cell 1 has the potential to divide. So again, the cells will increase. On one side, the number of cells are increasing, and on the other cell, the function is performing. So maybe the other stem cells express another gene, like the gene for the hemoglobin. So if this cells, this is the daughter cell 1 of the previous one, and it expresses a gene for the hemoglobin. So hemoglobin is a protein, and protein is formed from polypeptide. So there also will be a gene for the hemoglobin. Suppose the gene for the hemoglobin is gene F. If it is gene F, and this gene F is expressed here, so of course it will remain dormant here. It cannot multiply here because it's acting as a stem cell, but it will be expressed here, and now this cell will make the hemoglobin, so it will become specialized as RBC. Now it's converted to RBC, and RBC cannot be further multiplied, so it's become specialized. And again, these cells will be remain, all the genes will not be expressed, so it again acting as a stem cell. So it again divide into two more cells with the same procedure. It will be repeating again and again. So at one side we are producing the specialized cells, and on the other cells we are producing the stem cells. So one stem cells produce other stem cells and also produce the specialized cells. So specialized cells are actually originated from the stem cells. Once their genes become switch on, their genes become expressed, and they form a particular product. So whenever the examiner asks you how a cell becomes specialized, so the answer will be once a particular gene in a stem cell is expressed, it is transcribed and translates and produce the desired protein, which is related to the function of that cell. Then the cell is said to be specialized. But once the gene remain unexpressed and the cells do not produce any special type of protein, but they just have the potential to divide again and again, then it is called a stem cell. This is the difference between the stem cells and specialized cell. So this is the difference between the stem cells and specialized cell. The name of this chapter is cell cycle, So cell cycle means repetition of the cells to produce identical cells in a series of events. So what do you think? What kind of cell we are dealing? Specialized or stem cell? Of course, we are dealing with the stem cell because stem cells have the potential to produce identical cells. We are not talking about the specialized cells. In the previous grades, like in the O level, you have been told so many types of cells like the red blood cells, white blood cells, root hair cells, neuron, and all these cells were specialized cells. Even the sperm cells, the egg cells, they are the specialized cells because you even didn't study that one sperm cells produce other sperm cells. Sperm cells do not have the potential to produce more sperm cells, but it just have to perform its function and then it will be disintegrated. Similarly, RBC cannot produce other RBCs. When the cells become RBC, then of course it will be died, disintegrated, and new RBC will be formed from the stem cell. Similarly, a neuron is a specialized cell, so neuron cannot produce more neurons, but they will be replaced from the stem cells. So, of course, in this chapter we are dealing with the stem cells only, and for the entire process of cell cycle, we generally have three important steps in the entire events which is going on in the process of cell cycle. First one is called interphase, then mitosis, and then cytokinesis. So there are three important steps in the cell cycle. Now, if you talk about the interphase means between two phases. Inter means between and phase means stage. So interphase is actually a phase which takes place between the two consecutive cycles. Once a cell is completely divided, and after division, when the cells become into two daughter cells, like if this is one of the cell, and it has been divided into two daughter cells. Now you know that these cells also have to divide into two cells. Each of one of become the stem cell. Now, once these cells undergo into two daughter cells, first it must have to grow. These cells must have to grow, increase the size. The nucleus also become increase, and now it has the potential to divide into two. Similarly, these cells also grow first of all into a bigger cells, and then it is divided into two cells. This stage, I mean, where I'm telling you this stage and this stage. These are actually interphase between two cell cycles. One cycle is completed here and the other cycle is starting here. So the phase between the two is called interphase. This is interphase. Now what's going on in the interphase? I mean, it just prepare the cell, prepare the daughter cell, prepare the daughter cell. Prepare the daughter cell for the next cell cycle, cell division, for the next cell division. Prepare the daughter cells for the next cell division. So it means it is also called a preparatory step. A preparatory step. It involves to grow the newly formed cells to grow. The contents will be doubled because once the contents of cytoplasm, if we have 100 contents of cytoplasm nanogram, so each of the daughter cell will have 50 nanogram of the cytoplasm. I mean half. But the original cells contain 100. So if you want to divide the newly daughter cells, now the cytoplasm will be further reduced to 25 nanogram and then 12.5. So at the end, the cytoplasm will be finished. So what should we do? Now, these young cells which have 50 nanogram first make the cytoplasm doubled again to 100 nanogram. The organelles should also be doubled because each of the daughter cells have half of the number of organelles. So doubling of the organelle must be taking place in the interphase. The cytoplasmic contents should be doubled. The DNA should be doubled so that after division, the original number of each of the organelle and the DNA should be restored. This is all taking place in the interphase. So interphase is just the preparatory step, and it enable the young daughter cell to become grow and should be acting as new stem cells for the next cell cycle. So it should be ready for the next cell cycle. So it's two consecutive cycles. In between the two cycles, there should be a preparatory step which is called interphase. So interphase is further classified into three steps: G1 phase, S phase, and then G2 phase. So we have three steps in the interphase: G1 phase, S phase, and G2 phase. Now, these three things are very important because once you want to grow the cell, I mean, if the daughter cell or the young cell, which is just formed now from the process of cell cycle, should be grown. Daughter cell or the young cell should be grown. That's why we use the word G1 for gap or growth phase. Now, there are two important things in the cell to be increased. Once the contents of the cytoplasm, I mean the doubling of the organelles, if there are 1000 of ribosome in the daughter cell, it must be 2000 now, become doubled, so that once it's again divide, so the origin number should be restored. The mitochondria, the endoplasmic reticulum, the Golgi body, the lysosomes, the vesicles, all these things should be doubled in interphase, and that's in the G1 phase. And then the S phase is related to the doubling of DNA contents. The contents inside the nucleus should also be doubled, and that takes place in the S phase. Similarly, G2 phase is related to the formation of certain important protein molecule. So it means if you are concerned to protein formation, then you must have to undergo the process of transcription and translation to produce different kinds of protein. What kind of protein? Because we need certain types of enzyme in the process of cell cycle, and enzymes are protein, and protein to be formed by the process of transcription and translation. And from transcription and translation, we need certain genes to be expressed. So even in the G1 phase and G2 phase, some protein molecules are also formed. So here we also have the process of transcription and translation to generate some of the protein contents, along with the doubling of the organelle and cytoplasmic contents. Even if you're talking about the mitochondria, if you want to increase the number of mitochondria, so to form a newly mitochondria, mitochondria is also made up of protein components. The membrane is made up of protein. The protein channels are there in the membrane. We have electron transport chain in the mitochondria, the crista, the inner membrane. And all these are made up of protein. And for that protein, we need the process of transcription and translation, obviously. So anyway, there are three important steps in the interphase: G1, G2, and then S phase. Similarly, once the contents become doubled, I mean, if we have a cell here, this is the nucleus, and here the cell, complete cell. I mean, we have here 46 DNA, and suppose we have the cytoplasm here, and we have some, mitochondria here. Let's suppose here the mitochondria. So this is the mitochondria. Similarly, we have some other structure like the Golgi body. So we have some Golgi body molecule. We have the Golgi body. I'm so sorry, Golgi body structure, not molecules. Golgi bodies are organelles. Similarly, we have some structures like the lysosomes. These are the lysosomes. So now, if we want to divide these cells into two cells, so initially, if you want to divide it into two cells, then of course the daughter cells will have half the contents of these presents in the cell. I mean, if you just have to divide these cells into two cells, Then each cell, let's suppose this is one of the cell formed here, and this is the nucleus. Here, the other cell. Here's the nucleus, and here the cytoplasm contents. So if here is 46 DNA, then here should be 23, and then again 23. Here the mitochondria will also be in decrease in number. How many mitochondria we have? Here we have the mitochondria as 1, 2, 3, 4, 5. Oops. 1, 2, 3, 4, 5, and 6. So we have three mitochondria here. Like here we have the mitochondria. So we have the mitochondria 1, then 2, and then 3, 1, 2, and then 3. So we have 3 mitochondria, 3 here. And similarly, all the contents will be reduced to half. But this is not the case actually, because if the same case remains continue, then everything will be decreases in their concentration. So what's going on that at this stage here, what's going on that there should be one step where the cells will become increase in size. The nucleus will also be increased in size. Now here we have the DNA as 92 DNA, not 46. It becomes doubled. The cytoplasm should also be doubled. The mitochondria here should be 12. Instead of 6, we have 12 mitochondria here. So mitochondria will be 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Similarly, the ribosomes will also be increased in number. Here we have the ribosomes. How many? 1, 2, 3, 4, 5, 6, 7. These are lysosomes, sorry, not ribosomes. So if they are 7, so here its number will also be increased to 14. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and 14. So 14. Similarly, the Golgi body will become to 8 and doubling. And once it again divide, the same number will be restored here. Restore. And here doubling. Here is the doubling. Now this step is called interphase. Doubling. And this step here, this one is called the cell division. Once you talk about the cell division means nuclear division, division of the nucleus, and the second one is called cytoplasmic division. Not only the DNA will be divided, but also the cytoplasm will be divided. So cytoplasmic division as well. So nuclear division is called mitosis, and cytoplasmic division is called cytokinesis. It means at the time of cell division, you say that usually we misinterpret that mitosis is a kind of cell division. No, mitosis is not a kind of cell division. Mitosis is a kind of nuclear division. 2 identical nuclei are formed with the same number of DNA contents at the end of mitosis. If we have 46 DNA, so at the end of mitosis, we have only one cell, but we have 2 nuclei in one cell. That's the end of mitosis. Because if you have just only one cell with two nuclei, that's the end of mitosis, because we divided the nucleus, not the cytoplasm. And once the plasma membrane is also divided, then we say that the division of the cytoplasm as well. Like if suppose this is one of the cell, and as a result of nuclear division, we just got two nuclei, and both the nuclei have the same number of DNA. So we have this 4 DNA here and 4 DNA here. It means that this cell is somewhat a bigger cell, as I said, 2 nuclei, but the DNA content has been divided into 2. The overall number of chromosomes in this cell at this time is the DNA are 8 DNA molecule here. 8 DNA molecule in total. But once this cell is divided, like the plasma membrane is divided in this way, makes an invagination inside, and this part remains to be disappeared. The membrane is like pinched off inside. And then this membrane is going to be deeper and deeper, growing deeper and deeper. Same way. And finally, the membrane will fuse together, like in the same way as here. They will going again, moving inside. And finally, they will break up the two cells, and the cells will be separated from one another. So this cell has been moved out, and each cell have now one nucleus. And of course, the cytoplasm is also divided now. Initially, they were the single cell have the double content of cytoplasm, and now the cytoplasm is also divided into 2. Of course, the organelle should also be divided here, and the organelle should also be divided from in equal number from both the side. And similarly, the other organelles like the Golgi body and the endoplasmic reticulum, that should also be divided into equal number. This is called cytokinesis, and the division of the nuclei is called mitosis, while the cytoplasmic division and the organelle division is called the cytokinesis. So overall, we say that in the first step, there should be doubling of the DNA, doubling of the cytoplasmic contents, and this is called the interphase. And then the division of the DNA first by the process of mitosis, and then division of the cytoplasmic content. So initially, both of these contents will be doubled, like DNA is also doubled and the cytoplasmic contents are also doubled. But later on, each of them become again divided to the original number will be restored. Now that's why we see that they are the same thing. Like here, if we focus on the process of cell cycle, as I told you, and we say that the cell cycle comprises 3 important steps. What is mitosis? Let's talk about the mitosis now. Mitosis is a step of cell division. It says mitosis is a step of cell division, not a type of cell division. Mitosis is a step. There are 2 steps in the cell division. One is called mitosis, and the other one is called cytokinesis. Cytokinesis, as I told you. Cytokinesis and mitosis are 2 different things. Mitosis is a nuclear division. Nuclear division. It divides nucleus. So DNA contents divides equally. And one cell is made up of 2 one cell is made up of 2 nuclei, each have the same number of DNA contents in mitosis. But what's going on in the cytokinesis, then division of the cytoplasm? So here, precisely a nuclear division, which results in all the daughter cells being produced with the same number of chromosomes. Because once the chromosome number have been doubled, you double the chromosome number. So once you become half it, then again the same number of DNA will be restored as before the doubling as the parent cell. The ability of mitosis to produce daughter cell increase the number of cells needed to allow growth to occur in multicellular organism. We are all multicellular organism. How our body grow? Because the number of cells are increasing in our body. So how the cell will be increasing? Of course, by the process of cell cycle, the number of cells is going to be increase, and we are growing. Our body parts are also growing. And some of the cells are lost from the body. They will be replaced by the cell division of mitosis. As all the daughter cells are identical to the parent cell, this is essential for growth in tissues so that the new tissue cells are exact copy of the existing cells. Otherwise, tissue rejection will take place, or the cells will encounter with one another if they are different from one another. It's the same story like if there is one of the policemen in between the students in a classroom. So the policeman is quite prominent as it has a youth police uniform, and the rest of the students they have the school college uniform. So it's quite prominent among the students. So if they are all have the same function and they are the same structure, then there will be no encounter. So it is necessary, if you want to increase the number of cells and all the cells are exactly the same, they perform the same function because they work collaboratively with one another, then they must be produced by the process of mitosis. But if you make some cells different from the other, then of course they will interact with one another and an immune system will be triggered. That's why it's important for mitosis to produce identical cells. Otherwise, it may results in the immune system to be triggered. Now, in mitosis, before mitosis, there should be interphase. Initially, the DNA should be doubled. DNA replication occurs during the synthesis phase of interphase. I told you that in the interphase, there are 3 steps: G1, G2, and S phase. S is for synthesis. So the doubling of DNA is taking place in the synthesis phase or S phase. Then what's going on in the G1 phase and G2 phase? I'll tell you what's going on in G1 and G2. But the DNA doubling should be taking place in the S phase or synthesis phase in order to double the amount of DNA in the nucleus of the parent cell. During the DNA replication, all the genes are copied exactly the same to produce another strand of DNA. And I told you in the previous chapter about the semi-conservative replication of DNA, how a DNA is copied to be by the process of replication. I told you. So the same thing is going on. If you want to increase the number of chromosomes from 46 to 92, so each chromosome is made up of one DNA molecule. So the DNA should be split open into single stranded, and they will be copy the identical. They will make the free polynucleotide strand. They will make the new polynucleotide strand from the free nucleotide in the nucleus, and the DNA content will be doubled. This ensures that all the genetic information is retained during the doubling of the DNA. As I told you that the daughter cell will be exactly the same as the parent cell. So if the parent cell have 1,000 genes, so each and every gene should be copied in the daughter cells. Otherwise, some of the characteristics in the phenotypes will be missing in the daughter cells. That's why to ensure that each and every gene should be copied, the DNA molecule should be doubled in each cell. So that should be replicated later on. Now, no genetic variation occurs in mitosis, because if there's variation, it will be encountered in the immune system. So that the chromosomes in each daughter cells carry the same hereditary information as the parent cell. This helps to maintain genetic stability. Mitosis maintain genetic stability, no variation. But meiosis is another type of cell division, another type of nuclear division which maintain genetic variation, because in meiosis variation occurring, but in mitosis there's no variation to be occur. Now, maintained in the offspring. This is due to the random distribution of chromosomes which contain identical gene, as the DNA content of each chromatid is the same as its sister chromatid. So once the DNA molecule, as I told you, like this is one of the DNA molecule with the two strand. Now let me draw it some more elaborately. If this is one of the DNA molecule, and both the strands have the complementary base pairing system, so when they separated from one another and each one makes the new copy. Now both of these molecules will be identical to one another. Both of these are identical to one another, so there will be no variation in the DNA molecule. That's why we say this is due to the random distribution of chromosomes which contain identical genes. And this also enable organism to asexually reproduce and have offspring with identical gene as the parent. So there are some organisms which are produced by the process of mitosis, by asexual multiplication. They are not going to fuse their gametes with the male and female gametes, but they just produce the identical cell. And they produce the new daughter cells, but they are exactly the same as the parent cell. So in this case, all the genes should be copied, so they must have the same morphological and physiological characteristics as the parent cell. Variation in DNA of the same tissue produced by mitosis may result in tissue rejection as well, which may encounter your immune system, as I told you. Now, I told you what is the interphase. The process of mitosis is very complex. However, for simplicity, it is divided into prophase, metaphase, anaphase, and telophase. There are 4 steps in the process. As I told you, like here, the interphase is made up of 3 steps. Similarly, mitosis is made up of 4 steps. That's prophase, metaphase, anaphase, and then telophase. So each of one have a different time period for how long the process remains continue. Now, in this case, the longest phase in the cell cycle is the interphase, as interphase occupy 80% of the total time for one cell to divide into 2. So a cell to complete the cycle, it takes 80% of the time in the interphase, one of the longest phase. 17% is about in mitosis, and 3% is only in cytokinesis, a short time period. And in 17 phases, the prophase, metaphase, and anaphase and telophase collectively makes the 17% of the mitosis. So if you make the pie chart here, like in this way, let me make some different. Sorry. Now, in this flow chart, if we just have to divide, it's like in this way. Now you can see that this is the interphase. This is the interphase. And now here is the mitosis. Here's the mitosis. And the rest of the part, this is cytokinesis. Oh, sorry. So it should be like to remove. Yeah. So it like be this one. Right. This is the cytokinesis. And the process of prophase is also of 3 steps. So here we have to divide like this one. 3 steps. So we have this is the G1, which is the longest phase, and then the S phase, and then the G2 phase. And this is the mitosis, and here is the cytokinesis. Cytokinesis, the entire process. So let's talk about here the process, G1 phase, and so on. Here they have given some time period for the divisions to be occurred, like prophase, interphase occupy about 80% of the time, in which G1 phase occupy about 35% of the 80%. And similarly, S phase occupy 25% of the 80%, and G2 is then 20% of the 80%. Mitosis 17%, of which 6% is the prophase, 4% is the metaphase, 3% is the anaphase, and again 4% is the telophase, and the 3% is cytokinesis. So if you just count this one, that makes the 17%. And if you just count this one too, and along with this one, so 35 plus 25 and then plus 20, it makes 80%. And of course, this is the 3% remaining one. This is the contributions of the entire process. So we will continue it in the next video about the process of mitosis and interphase and cytokinesis in some more detail. Stay with us. See you in the next video.